guys, today we're going to be talking about topics 2.5 and 8.1, which discuss enzymes. So remember that there's tons and tons, thousands of chemical reactions that take place within our cells. This is called metabolism. These can be both building up reactions, anabolism, or breaking down, catabolism. And in order for these reactions to take place under normal pH and normal temperature, they need a little help from biological catalysts or enzymes. Before we begin, I want you to take a minute and read understandings 2.5.1 to 2.5.2 and 8.1.1 to 8.1.2. These are the understandings that this video will cover. In addition, I'd like to challenge you to try and do kind of something that models an enzyme-related reaction. So I want you to change one letter at a time from the word tread to get to the word blink, and every word in between must be a real English word. So try that out. Okay, so we've already discussed metabolism to some extent. Remember, like I said, it has anabolic and catabolic reactions. Anabolic is building up, catabolic is breaking down. And metabolism can consist of two types of enzyme-catalyzed reactions. It can occur in a linear sequence, like the one that you see pictured here, or it can be cyclic, which we'll see a little bit later. In the case of a linear reaction, basically what we have is a starting substrate, an initial enzyme, an intermediate substrate, another enzyme, intermediate enzyme, and then finally our final product. So this linear sequence can vary in length and the number of intermediate steps and the number of enzymes. So again, I just want to reiterate that enzymes are biological catalysts. They are composed of protein, and they make reactions possible in living organisms. Normally, if you were to do a reaction in a lab or an industrial setting, you would need one of the following: extreme temperatures, extreme pressures, extreme pH, or a high concentration of the reacting molecule. When we're talking about living organisms, usually there are none of these things going on. Usually it's a normal temperature, normal pH, normal pressure, and reactants are present in low concentration. Therefore, they need a little bit of help. Enzymes are very effective even in small amounts. It doesn't take a lot, and the main reason for this is that they remain unchanged at the end of the reaction. So enzymes can be used over and over and over again in a living organism. Enzymes work by lowering something called activation energy. Now, no matter what, even if the product is lower energy than the reactant, there still has to be an initial input of energy to kind of get that ball rolling, to get the reaction started. And so what an enzyme does is it greatly decreases this amount of initial energy in order to decrease the overall amount of energy needed and to get the reaction started. So here we see a graph for the amount of energy needed and the progress of the reaction, and we can see without the enzyme and with the enzyme. And so the amount of energy needed with the enzyme is much, much less, allowing the reaction to happen under normal living conditions. Alright, every enzyme has what's called an active site. The active site is the site at which the substrate or starting material enters. It attaches to the active site. The enzyme decreases that activation energy, taking the substrate to the intermediate stage and producing the product. So that's what's going to come out of this reaction. And then the product is released and the enzyme is unchanged and it will re-enter this cycle. Every enzyme has a reaction-specific activation site. So enzymes can only catalyze one type of reaction or maybe a very small group of similar reactions. Therefore, the active site on an enzyme is specific to the substrate that it works on. This is the lock and key hypothesis, where the substrate acts as the key, and the enzyme has the lock. And so when the substrate fits into that lock, it can then produce the product. In addition to the lock and key hypothesis, there's also the induced fit model. This is where, again, the substrate matches the active site, but in this case, the enzyme is actually going to temporarily change shape in order to lock that substrate in place and form the intermediate molecule or the transitional site. During the transitional phase, other amino acids that make up the enzyme are going to be working on that substrate, breaking specific bonds within the substrate to form the product. So in the induced fit model, like I said, that enzyme is going to have a temporary shape change to allow the interactions that are needed, and then it will release the products to do the reaction again. And I should note here that enzymes don't always work to break down larger substrates. They could also be building up, so combining two substrates to form the product. Like I mentioned at the beginning of the video, we've got kind of two types of pathways. First is linear. So in a linear chain, the product of one reaction, here we go, becomes the substrate of the next. Each intermediate step has its own enzyme and we go down the row until we reach the final end product. The other possibility is a cyclic pathway. In this case, this is going to be a continuous cycle that's occurring constantly, really. And so we've got the initial substrate that enters the cycle at some point. Again, each intermediate becomes the substrate for the next step, and it goes around and around. At some point, the product exits. However, part of the product exits, part of it becomes intermediate 3 and continues through this cycle. And so this, like I said, is a never-ending cycle that keeps going around and around and around to produce the needed product. Now, in living organisms, the attraction between an enzyme and its substrate is very, very weak. They're not chemically really attracted. They largely come together randomly, and so it's due to random movement of the enzyme and substrate within the aqueous solution of the organism that allows this reaction to happen. So this is why it's advantageous for living organisms to be largely aqueous in their internal environment, as well as in constant motion, in order to bring these enzyme-substrate combos together. Enzymes are the topic of a lot of study today, especially as we work to find new ways to fight disease. There is a branch of research called bioinformatics, and this is where research groups can add information to a large and growing database to allow other groups to access their information, thus decreasing the amount of time it takes to make progress. So this has given rise to efficient research of metabolic pathways. So there are massive libraries that exist of chemicals that are tested individually and allows scientists to identify and know about a range of target sites, products, substrates, etc. So one example of this being used is to treat malaria cases that are resistant to anti-malarial drugs. So remember, malaria is a disease that's caused by a pathogen, a plasmodium, that requires both a mosquito and human host to complete its life cycle. And bioinformatics has been used to test and catalog over 300,000 chemicals that were screened on a drug-resistant strain of this plasmodium. And because of this research, 19 new chemicals have been identified to inhibit enzymes that normally an anti-malarial drug would inhibit. And in addition, 15 chemicals were identified that will bind to proteins to help scientists locate, or medical professionals, to help scientists or medical professionals locate the plasmodium. So pretty interesting stuff, a big newer area of research that will be very useful as our fight against disease changes over time. Please write down any questions that you have, and we will chat more in class. Have a great day. Bye.